It's important to note that enzyme activity is regulated, meaning that enzymes are not always active all the time. And that's important because the metabolic needs of cells change over time. So, for example, during development, if we look at the kind of the developmental time known as puberty, we know that there are certain cells that become more active, and those cells start secreting hormones that are involved in the kind of the process of puberty. And so these were enzymes that were maybe turned off or not active earlier in life, but then they become activated during that time known as puberty. Different cells in the body also have different metabolic needs. So some cells need to have some activated enzymes, whereas other cells might not need those same enzymes to be activated. And so there has to be a way in order to turn on enzymes when they're needed and turn them off when they're not needed. So let's start by taking a look at competitive inhibition. So competitive inhibition is when a molecule other than the substrate is able to bind to the active site. And when that molecule binds to the active site of the enzyme, it blocks the substrate from binding to the active site, and therefore the enzyme is not able to catalyze the reaction. So this top reaction that we see up here, this is the normal reaction. And you see we have the enzyme here as this kind of gray Pac-Man looking thing. We have our substrate up here. And when the substrate binds to the active site, it causes the substrate to break into two pieces. So basically the substrate is actually made up of these two pieces here that get separated by the enzyme. Now, in competitive inhibition, that's what we're seeing down here at the bottom, where we have something that looks kind of like the substrate. So it still has this a similar shape as the substrate, but it's not the substrate. And what happens is that it binds to the active site and it physically blocks the active site. So now the substrate can't get to where it needs to go, which means that the enzyme can no longer catalyze that reaction because the substrate can't get to the active site. Non-competitive inhibition has the same outcome as competitive inhibition in that the enzyme is not able to access the substrate to catalyze the reaction. However, it's accomplished in a slightly different way. So non-competitive inhibition occurs when a molecule other than the substrate binds to an enzyme at a location that's not the active site, but it still blocks the substrate from binding to the active site. So when we say a molecule other than the substrate, this would be our inhibitor. And it's going to bind to the enzyme. It's just going to bind to it in a spot that is not the active site. So once again, in this picture down here, the top one is showing us a normal reaction where we have the substrate fitting into the active site of the enzyme, and the enzyme catalyzes the reaction to break that substrate down into its two pieces. In the bottom one, we see the inhibitor. In this case, the inhibitor is not binding to the active site, but it's binding to a secondary site. But the result is the same because, as you can see here, it's now blocking the active site. So the substrate can't get to the active site, which means that the enzyme cannot break down the substrate. Another form of enzyme inhibition that I would like to talk to you about is feedback inhibition. And feedback inhibition is kind of like a self-regulating reaction. So remember that a reaction can be written as a reactant, and then an arrow, and then if there's an enzyme involved, you write the name of the enzyme above the arrow, and then that gives you the product. Now remember, the reactant is also known as the substrate. Well, in feedback inhibition, what happens is that the product becomes the inhibitor. So you can see in this picture at the bottom that we have a substrate that binds with an enzyme. And that produces a product, which in this case, that product actually becomes the next substrate. So this is like substrate number 2. And that substrate binds with a second enzyme. So we'll call this one enzyme 1 and this one enzyme 2. And when that substrate binds with that enzyme, it gives us this product, which then becomes the next intermediate substrate. So this is like our third substrate. And when that substrate binds with our third enzyme, it produces this product here. And this product ultimately becomes the inhibitor. And so it can actually bind to the original enzyme 1 and kind of shut it down. So this would be an example of like an allosteric inhibitor because it changes the shape of the active site. But the key thing, regardless of whether it is an allosteric or another form of non-competitive inhibition, the key thing is just that the product becomes the inhibitor. And so what this means is that as more and more product is being made, more and more of the enzymes are going to be kind of turned off or down-regulated. And so that's why I said this is kind of a self-regulating reaction. It's because the product actually shuts down the reaction when it starts to get too concentrated.